the third supervisor in the last 4 or 5 years. So these bright young people always find great opportunities to move on and to do bigger and better things. And all I can say was it's great to have them while we do and take advantage. So the way it's going to work today, just with this presentation, Phil's going to give you a narrative rundown of what happened on the lake this year, what he found, what the crew found. And then when he's finished, he'll field any questions about actual work on the lake. And then if anybody has any further questions, particularly I expect that people would want to know what the status update of the removal, the biocontrol is. I can follow up with that after those so welcome, Phil Mack.
Hello. Oh, we got a couple pages here to go through, so it shouldn't take too much time. Anyway, my name is Phil Mackey. Many of you might not know me, but I've been the dive supervisor in the control program this year and the last program last year. So I've got a little bit of time to be supervisor in my belt here. Let's see, I've been a crew member since 2012, where I was just hired on as a field assistant, and that was the first year that there were 2 crews running the lake. So we had a full crew of 8 people that year. This year again, we had full crew of 8 divers, 6 turning divers, and then we had 2 brand new divers. The 2 new divers were guys only. They each brought their own good qualities to the group. And we had one that was a local boat access resident, and another university student that came from Ontario. We managed to hire all over Canada. That worked out pretty nice for divers looking for work. So we spent a couple minutes giving you some observed changes that I have witnessed since 2012 when I first started. 2012, that was the first year that we had this cruise again, and the shorelines were in bad shape. The South End Bay was completely out of control, and as a crew, we spent just weeks and weeks plowing through the South End along the South Shore, pulling hundreds of oil plants, using truckloads for turning all these plants to the what was that place called? Durans, yes. I'm sure if anybody's collected any of the topsoil, it's mostly comes from the lake. Milfoil was nearly the only plant that was populating the shorelines in the south. Overrun, completely overrun. And that was the first year, the 2 crews, 7 days a week control program. We have steadily regained control over the entire shoreline of the lake. So things are looking good. Native plants such as the Canadian and large pondweed have regained their original habitats. Basically, blood all milfoil. There's nothing left, and it's fresh topsoil. Sort of an analogy for gardening. And so all these native plants can come in and repopulate, which is excellent. And that also competes with the milfoil trying to regrow. As we all know, milfoil is very good at establishing itself. So 2015 control year was a continuation of the successes that we've seen since the 2012 dual crew program started. If I were to sum up 2015 in one word, it would be progress. This season, with its 2 crews, we are able to push into new areas that have not been controlled before. All the buoyed off areas in the North End Lagoon, which is highly protected. We were able to do many days in there, multiple days in the North End Lagoon. This has vast swaths of milfoil plants. Because no one really can go in there, you can't really see, but there's large areas of very tall, mature milfoil plants with full developed root structures and so on and so forth. But not only milfoil has large areas of native plants like water marigold, very large actually. They grow up to about a meter in length at that North End Lagoon, and water shield, which is closer to the shore, and it's a native plant in the area. Yeah, okay, good. And coontail came too. But it is hoped that in controlling the lagoon will slow the proliferation of fragments and coast properties, salmon properties have in the past and during this year as well at patches that we've had to control throughout the year. We've also progressed down Christina Creek. It seems kind of different that we do that, but at this point it makes excellent sense. There's a lot of boat access and boaters that go down there. The sole drain to Christina Lake. That's where all fragments are going to flow down and root in the soil. And Christina Creek is really shallow. We're talking 4 feet at low water levels. And it's easy for any of the boats to chop up any of these fragments of milk oil attached to the boat, and then they can go out around the lake, and we're going to have proliferation of milk oil. Once again, as a crew, also found in newly discovered areas southwest, so west of the public beach, around Silver Birch. There's a large swamp, large patch of milk oil that we located out there, and managed to bring under control. It was never been controlled before. It was far from shore because of the nature of the depth of the water there, 5, 6 feet. But we're talking maybe 100, 150 meters from shore. So, yeah, it's a large literal zone that we managed to find fairly far up from shore. And so, we have 575 recognized sites that we have numbered and categorized as oil plant levels around the lake. And there was, in 2011, at the end of the year, our good supervisor, Andy Gilmore, I'm sure you all remember him, he had done a full plant count survey categorizing each of the 535 sites with low, medium to high oil plant counts. And each one of those 575 sites has been reduced from high to medium, medium to low, and low to continually being low, as far as plant counts are concerned. So we've had an excellent impact on the shoreline of the state. So 2015, let's check out for next year, 2016. Plans for 2016 will again focus the areas of dense milk oil plants will be situated in the South Bay. It seems that the high amount of population, low traffic, and the shallow waters just makes it a breeding ground for milk oil and midwest too. It is hoped that the crew will be able to make an impact on the protected nature park between Sutherland and Christina Creek. This is the buoyed off area that's during the summer. Well, we got some excitement. All right, yeah. We haven't did that, but with the 3 times that we've installed in the spring and fall, those are large patches of oil and large patches of all the other plants as well that are needed for the fragrant water. That's one that seems to be coming up quite a bit. Fragrant water. Oh, she wants one. Okay. All right. All right, we'll do. Put it on the list. Yeah, that seems to be it for short little presentation for anything. So, if anybody has any questions on milk oil, etc., we're in ahead.
You may not have questions, but a round of applause.
Yeah. So, in terms of removing large volumes of and this is supposed to be for composting rate, we have an idea of the amount of nutrient removal that maybe an idea of how much nitrogen and phosphorus will be in the truckload that they'll want, right? And wondering if something that we measure and
Interesting question. I don't know if I'm qualified to answer that.
What, don't drink the running garden?
Oh, yeah. Yeah, absolutely.
All right.
Truckloads, let's take an example. Lakeside Resort, they have a huge beach. On a typical day where all 8 members are at, we'll do like 3 or 4 truckloads before the lake. There's a lot of weed, and it's not just them, it's the root structures and the beach up there as well. And it's a lot of water as well. So, the plants, when they dry out, they lay nothing. So they're very wet. Yeah. So I've just handed this. Is the Christina Stewardship Society Underwater World. It is a little booklet showing all
If nobody's seen this yet, most people have. If you want to learn about our native aquatic species and invasive species, this was written in 2013, but it includes the newest one, which is Kirby Ponzi. Thank you, so awesome. And so you can learn new plants, so you can see what ones are good and do that. So if you're in development, you'll see a lot of areas with native plants that are filling in where milfoil is in the shallows, right? And over there.
Okay.
right. All right. More questions? Yes.
Opposite the meter part, there is the forest of water.
Yes.
How do the divers get in the forest with mineral in the
With great difficulty. We are able to run the water here.
the water lilies? The great water lilies are going to there's no way that that's needed. Especially have to look at photos of that to see if they're identifying. But the ones that you're talking about, there's also a lot of the water field there. Yes, in the same. That might be what you're looking at. So that's part of what's happening. Yeah. So those are the problem.
For one answer. All right. And off to Alan.
Thanks, Sean. I think the crews are doing a really good job. We've been hearing a lot of positive comments, a lot of lakefront owners and comments that things have never been better. But again, it's a lot of work and it's an ongoing commitment of resources. So one of the things that we've been looking at, and this has been going back to 2010, 2011, we're looking at a biocontrol agent for the weevil. There were a few bioscience on the states that was plans that they do a weed to eat the milfoil and compromise it and to be an effective control program. We always felt that that would be a good complementary program to the they could handle the less dense areas that we would never have any chance of working with, and then we would potentially be in the south end of the lake and working with some of those really dense patterns. So we continue meeting and brought the agencies together, basically asked who was required to use this legal in the lake. There's, I will say there's been no progress. We did receive, finally, after almost 5 years from our first request, this March 2009, we received an email from the province saying, okay, if you guys want to do the legal, this is what you have to do. And they laid out some detailed description of work, including the biocontrol agent, reported outcomes, source of biocontrol agent, quantification, monitoring protocols for targeted and non-targeted species, indication of support for the proposal and engagement with First Nation, which is all good stuff and all have to happen. But that is, we looked at this list and it is well beyond the scope of a small to do this kind of work for a biocontrol agent. We thought this was a bit. I'll be very brief. We thought this was a bit. Maybe there's a better way. It's 2 words. One starts with B. So this work, we biocontrol agents are not uncommon. We have some terrestrial control programs we use. We talked to Bart Stewart earlier today. We use about a dozen different biocontrol agents. Each one of those bugs is tested extensively by the province, by different agencies. The tests go on for years. They cost up $1 million a test. And basically what they do is, I just briefly read out to you. They do all that work. So they pretty much just shifted it. If you guys want to use it, you got to do all that work. If you're a chemical or something, you need to, get it something approved. And it's just absolutely on the scope. So on the political side, Reverend Greger has been, she has been battling this one. Every time she goes to a UBCM meeting, he gets in front of the minister to provide data, background, records of email correspondence, and trying to get somebody in the province to actually care about this. We feel that given the Okanagan from the Shuswap and Christina Lake, there's a large potential economic benefit from actually doing this work. But the people that run the programs for biocontrol, again, it's a common theme with the province. They're completely understaffed. They are under-resourced. They had a long-term fellow there that was there for 40 years, suddenly retired. At last count, nothing in place. It doesn't seem to be on to replace it. According to Lisa, they have to do a business case to replace an existing program, even though it's foresighted. So it's not good. So that leads us to what are we going to do with this program? The sectors are working really well. Every year, everything's on the data. We look at the crew size, the crew composition. What we're running into now is things like, equipment, aging equipment. That I think we're going to continue. We have to look at some capital investments. It's a big challenge. And the point that Graham made about what is the nutrient removals. We do a lot of stuff. But it's pretty anecdotal. We look at stock stem length and stem diameters and stuff in areas that have been controlled. We're looking for its compromised abilities to regenerate. That said, the crews are busy picking, and we're not doing an actual scientific analysis program. It would be nice to have both opportunities to do it, but I've got the crews on the level of the so that's where we are.
So the one part of that I'll add to is I did meet with the actual minister this year. It took me 3 years to get one of those because they kept sending substitutes. And my message was maybe it would be a good idea to read the information. Number one. Number two, wouldn't it be nice if we got ahead of something and said behind it for change? And number three, when Christina Lake received a paper from you that said, so sorry, you've got the wrong label, and we had to send your own paper back saying, so sorry, we have the right label, that's sad. So that was the message that I gave the actual minister this year. We'll see what happens from there. I do have some other people working on this with me. Donna Barnett out of 100 miles is getting she really pushed the button. She got a little. So we've got extra people working on let's see what the answer is. Okay, having said that, let's go on. We're going to have a chat with Heather Layton. And Heather, where are you? There you are, hiding behind me. And Heather is hiding behind me. She's going to talk, she's actually the right-hand person to Brenda. And Heather's been with us for a few years now, so she knows Christina Lake pretty much inside and out. This is your second home or first home or when you have home. And so you're presenting information on areas of education and monitoring programs. Hands together for Heather Lake.
Thank you, everyone. So, yeah, I'm Heather. I've been with Stewardship Society now for 3 years. So this is actually my third Watershed review. I'm going to be talking a little bit about our AIS program. If people don't know what that is, Aquatic Invasive Species. So first I'm going to talk a little bit about some of the impacts. So one of the impacts is problems with the food web. So if mussels were to get into the lake, they would eat all the plankton that are in the water column, and it would cause a cascade of declining populations of all our fish. The second impact is cost to recreation and tourism. In the top right corner, beach has actually been closed because they applied herbicide to the lake to control Eurasian water milfoil, which is not allowed in Canada, only in the States. Another impact is displacement of native species. So in the bottom right-hand corner, you can see a crayfish, which is completely covered in zebra and tiger mussels. So that would eventually lead to the death of the crayfish. Lastly, economic costs. There was an estimate done that for each invasive species that is uncontrolled, it could cost $5 to $60 million per species by the year 2020, which is huge. Okay, so now I'm going to talk a little bit about the invasive species in our lake. So we just touched on the Eurasian water milfoil. So that's been being pulled since '86. The fragrant water lily, as we were also talking about. The brown bullhead, which is a catfish, and I don't know why somebody introduced this for a lake because nobody fishes for it. And the curly pondweed, another issue. We've noticed that it's starting to even get into the north end of the lake. So there's even patches that have been found at the north end of the lake. And it's starting to spread throughout the lake, even competing with the milfoil. The common carp, I'm sure we've all seen that. It's actually the third most frequently introduced species in the world. Hydrilla, it kind of looks like Canadian pondweed. I'll talk a little bit more about that later. The northern pike, this is actually a picture from Tech. They did a study on the northern pike in the Columbia River, and that was one that they caught by Robson, so not too far away. The snakehead, that was the one that got released into Burnaby Pond, and they actually had to drain the pond in order to get that guy out of there. The red-eared flyer is the pet we get at the pet store. So that's what kids will pick up at the pet store, and they actually live for 40 years. And I don't know too many kids that have their turtles for 40 years, but if they were to get into Christina Lake, they would compete with our native turtle population and could spread disease and all that kind of stuff. And then the American bullfrog. So these guys get huge. They can actually hop 6 meters, and they actually eat garter snakes, which actually prey on other species of frogs. So they could be a huge problem. And also lots of eggs that are really gross to other species, but not to eat them. So now I'm going to switch to our monitoring program. So this is our crew. There's Leo there. He's got a back. That's how we usually see him, shirtless and with fish in his hand. So then there's Holly Hexmer. She was our summer student this year. She did an awesome job. She was very passionate about AIS. She even messaged me and said, oh, what an impact this program has had on my life. She's going to all these conferences and stuff at her school at UBC in Vancouver. Ashley Lawrence was the Boundary Invasive Species summer student. So she was part of our this year because we did a project in conjunction with the Boundary Invasives. And then there's me underneath looking under the microscope. All right, so in our monitoring program, we have 2 different sampling methods. The first one is the plankton haul net. So that's that net there. So we'll take that out with him when he goes to do our water quality sampling. And he pulls it through the water column. And anything that's floating in the water collects in the bottom of this net. And then he puts it in a jar and brings it to me so I can look at it under the microscope. This is what we're looking for, is these guys right here. That's the veliger form of the zebra mussel. So they're very small, and usually we see a lot of stuff like this. These little critters and that kind of stuff. We also have the substrate samplers. So those are in 5 locations around the lake. We have one at Texas Creek, the RDK Depot Launch at Larson. We have one at Shoeley's. The Mary's residence, Dr. Mary's house, Christina Lakeside Resort. Oh, Shoeley's. So we did have one at Willow Beach actually this year, but it went missing, so we ended up switching it to Lakeside halfway through the summer. And these, the substrate samplers, they detect mussels when they already are starting to attach onto things. Whereas the plankton helmet is in an earlier form, so it's in larval form. So if you detect it in the larval form, you can stop it from spreading to other lakes probably quicker than if we find it on a substrate sampler, because by that time it's already a little too late. So no mussels found this year. We did have a little bit of a scare though. I'm not moving. No, okay. So up right here, that thing that looks like a muscle. So when I saw that under the microscope, my heart kind of stopped a little bit and I freaked out a bit. We have a microscope that has a digital camera component, so it records everything that we're seeing underneath the microscope. So when we saw this, we're like, oh, that kind of looks a little bit like a muscle. We were able to send this video down to the provincial specialist, and they confirmed that it was an ostracod. So it's a different kind of crustacean, it's totally native, and it actually is joined different, has different swimming patterns and stuff. So luckily, it wasn't a mussel, but because we were able to send this down, the provincial specialists were able to get their protocols in place in case they get any more sightings of mussels. So it was a good thing in the end, even though, yeah, it freaked me out a bit at first. All right, so now I'm going to switch into our educational program. So at the beginning of the year, new legislation went through about aquatic invasive species, and I think Barb's going to touch on that a little bit more after me. So we got a call from Chris Buckley at the border there at Cascade. He said that they were doing boat inspections and they had their decontamination unit from the roving team. So the two ladies in the middle, Jillian and Andrea, they were part of a roving team that got established this year. So we went down there, and this was their first blitz at the border to try and catch boats with zebra mussels. So that was pretty exciting. Also, we spent 3 days a week at the boat launch. So Holly and Ashley were there 2 days a week for half days down at Texas Point. And Simon Ryder, who is one of the milfoil divers actually, he volunteered with us this year. And he would go down with Holly on Saturdays. He was great to have on the team. He was very into aquatic invasive species, and he was always picking my brain about lake facts and that kind of stuff. So the data, get the numbers here. So of the 429 data entries we had, 24% of them were repeat visitors to the boat launch. Of the non-repeat visitors, 78% were from BC, 18% were from Alberta, 3% were from Washington. We had one boat from Alaska, one from the Northwest Territories, and 3 BC boats but Alberta residents. Of all the people that came and visited us, about 31% came and talked to us at the booth. So we keep track of everybody that's going through the boat launch. We are looking where they're coming from, what their license plate says, and all that kind of stuff. So out of all those people that were at the boat launch, 31% actually came up and talked to the people. Almost everyone that we did talk to, they said that their boat had only been in Christina Lake. But they were also really aware that if they are going to another water body, they're taking their boat there, that they make sure they clean everything properly, drain, the clean, drain, dry program, right? So it was really good to see some of the awareness from the first year that we were doing this program to this year. People are a lot more aware of aquatic species. Okay, our educational program continues. So this year we got a new banner attached to our wall. Just so people would see the sign and be like, oh, who is that? What are they doing? So we got one with our website on the bottom there just so people know where to go for some information. We also incorporate this into our youth program. There's a picture of me teaching the kids about fragrant water lilies down by the creek there. And then another initiative was the Don't Let It Loose program. So I think that's through ISCBC. So we printed out some of these stickers and we're giving them away to kids and telling them the importance of not letting your pets go into the lake or goldfish, turtles, all that kind of stuff, because we can cause all kinds of issues. Oh, and this is just a little thing at the end here I want to talk about. So we did get a lot of calls this year about native mussels. So people are like, oh, we found this mussel on our property. I think it's a zebra mussel. So I would go and it would end up being a native mussel. So this is one up here. That was, I think, Dan Romaine brought that one into us. And as you can see, it's a lot bigger than the zebra and quagga mussel. And also these guys, they bury themselves in the muck rather than attaching onto substrate. So just a little refresher, like these are the native mussels, these are the zebra and quagga mussels. I think it's awesome that we're getting these calls because people are actually aware of it and they're looking for these kinds of things on their own foreshore property. The other thing I want to talk about was native milfoil. So we have the non-native Eurasian water milfoil, which is in the top left corner. And then on the bottom here, we have northern water milfoil. So this is actually a native species, and we do have this in the lake as well. So the native milfoil, it actually has less leaflet pairs and it's a lot more stable. So you pull it out of the water, it kind of folds its shape, whereas the Eurasian water milfoil kind of falls apart in your hands a little bit. And then also, the picture I showed you of hydrilla kind of looks a little bit like Canadian pondweed, but Canadian pondweed is supposed to be here, and it's actually competing with the milfoil, so that's awesome. So just one thing, if you see as one and it has a whorl of 3, it's actually the Canadian pondweed, or also called Alodia. If it has whorls of more than 3, let us know and we'll check it out. So this little red-eared slider is wondering if anyone has any questions.
Yes, I think it's important for everybody to be aware also that watercraft includes stand-up paddleboard, kayak, canoes, other things are being kept at the boat launches. So, you know, just a point of reference for all of you, when these things come in, help us to spread the word to people who are bringing peace to the lake.
Yeah, another thing, yeah, but the roving teams, that was one of their comments, is that when people see the sign that says watercraft inspection, they don't clue in that that means canoes and kayaks and paddleboards and stuff. So even those things can spread, you know, these microscopic, you know, larval forms and stuff too. So, yeah. And fishing gear, exactly.
Any other questions?
After the video is dry,
Yeah.
how long can you do it?
They can actually survive out of water for 30 days, which is pretty crazy. And if you have any standing water, then they can survive even longer than that. So things like ballast water that might not necessarily become all the way dry, they can keep your muscles, no problem. Yes. I think so. I'm not too sure where their status is or anything right now, but they did get halfway through the year, they got more teams. So they doubled their amount of teams halfway through the year. It's the Ministry of Environment, so they're auxiliary conservation officers.
That's all I ever own.
Okay. Yeah. So,
and everything that they are planning to have to go through again next year. So things start earlier now. All the which, by the way, I don't happen to think is enough in my program.
yeah. Well, it's $2 million over 2 years. You know, when these mussels got into the Okanagan Basin, then it would be $43 million a year just to control them. So $2 million, in the grand scheme of things, is not a lot.
And when you have roving stations, you have a station there, people go through it, that station goes on to somewhere else, and a boat comes in. And that's all it is. What we need to do is follow the Idaho model and create a program that everybody research every year, and that's for the permanent station. So we're all pushing those questions. All right, thank you, Heather.
Good job, Heather.
Yeah, on the,
Not really. We did send them down to, our pictures down to Portland State University, the expert down there, and he confirmed the species. I can't remember the species name, but I think there's 4 different species in this area. But not really, we know where they are. They're at the creek, the mountains of the Cedar Creek. We've had some sightings, and then along West Lake, we've had lots of routes. Yeah, it's not the rock, it's not all that stuff.
Okay, onward we go, and carrying on with the same kind of conversation is, only this time on the ground, is our lead lady, Barb Stewart. Barb is the wealth of information.
Thank you, guys. We'll wait for Heather to get that brought up. So we, as Heather had mentioned, we do work quite closely with and really appreciate all the hard work that the Christina Lake Stewardship Society does on educating voters out in this area. It's so awesome to have them on the ground doing so much work. So I just want to touch on some highlights of our program for this year, for the Boundary Based Species Society, which focuses mainly on terrestrial plants, that is upland plants across the boundary area. But we also do some work on aquatic species, mainly outside the Christina Lake area. These guys are doing an amazing job here. We do an education program. You'll see some of the signs that are starting to go up at different boat launches, just trying to remind people of the whole Clean Grain Drive. Some of the work we've done out of Booth and Oxbow. It's great to control invasive plants, but sometimes you have to do a bit more to make sure your plant's going to be strong. So there's some restoration projects ongoing and planting, and there's Caddis there with his mom Jenny, and he's enjoying himself out in the cool weather, helping the plant stuff. And that's Wendy Hope from BC Parks, working hard out there in the cold weather. I wanted to talk just a bit about some of the priority species. As a region, we've come up with an operational plan of, what do we target? Invasive is an underfunded program, totally underfunded. So you have to make choices about what you're going to control and what you're not. So what we focused on is species that are fairly limited in distribution, that is fairly easy to control. So some of these species, the cutleaf evergreen blackberry that's out here at Christina Lake, 2 locations. Himalayan blackberry, also being controlled. Mouse-ear hawkweed, it's probably, I guess there's one other site out in Kokanee Creek Provincial Park, and then there's one here, and that's it for southern BC. So it's an important species to control. And of course, out in the community or in the provincial park. Long-spine sandbird, that's a really nasty grass, I guess you say, where the seeds just stick to everything and go right through your gloves. It's quite nasty. And it's out on the roadside out here in Christina. We've worked hard on this for the last 3 years, and we've had very little coming back this year. We think we're down at the point where if we keep on it for the next 3 or 4 years, we'll probably get rid of it entirely. Finless thistles, another species, there's about 3 sites out in the Christina Lake area here, and so we're actively hand-pulling those to not let them be spread. And Scotch broom, we're very fortunate, unlike the Nelson and Kootenays area where they have a lot of Scotch broom. We're very lucky here we don't, just a couple locations. So we're definitely not going to let that one get away. Of course, there may be still some in gardens, and we're going to work with educating gardeners and encouraging them to move it. Some of the species are a little bit more widespread, where we mainly focus on continuing work. Common tansy, there's quite a, it's coming, bleeding off of the Kootenays and coming this way. So that's something we've been working on controlling along roadside. Blueweed is really declining, at least along the highway roadside. There's still some private properties where it would be really nice to be able to tell people they have to control it. But at this point, we're still working on compliance. Borealism, which is definitely making its way through Christina Lake. Japanese knotweed, that's some of the patches, and it's great having the stewardship on the ground here because they're actually taking reports from landowners who are saying, hey, I think I've seen this weed, and then they're passing along to us to organize control for it. So it's a really good partnership. Yellow flag iris is one that we've done quite a bit of work on this year. So yellow flag iris is big, heavy, and it's hard to get out, and it is down Christina Creek. For the last 6 years, we've been controlling it in the community park here. And this year we started work down Christina Creek with some funds from Lisa Zesco's program with the Ministry of Forest Lands and Natural Resource Operations. So we started this year with digging and targeting. We've had really good success doing 3 or 4 years in a row with digging, but it's really heavy. This stuff, I mean, you're like 50 to 80 pound bags of iris, you're like dragging away from the creek, hauling from landfill. So this year there was some research being done. I was at Thompson Rivers University on tarping. So we started that this last fall, and we're going to be doing more of that down to Christina Creek. At this point, we're only doing control from the weir down to the wetland through District Lot 498, which is the Crown Lot. We hope that as we see success there, we'll be able to expand it down further. I just wanted to remind people about what some of our weed problems
used to look like. How many of you were here in 1971? I was. Anyone remember this? Yes, we had a bit of a weed problem. Next slide. And it didn't really get any better by 1984 either. Mostly diffuse snapweed. It was a big problem. The Ministry of Forest had started the biocontrol program, which Alan had mentioned. The province has always been the lead on biocontrol and pays most of the research. So I just wanted to show a slide of how things have changed over the years. Just to remind people that things have really changed. Our terrestrial invasive plants are we have far less of them now than we did in the past. The program really is working. We're actually getting grasslands recovering. And so we're actually starting to see our natural grasslands in their glory. So that's really nice to have that happen, and it's all across the entire boundary. We're seeing this change where we're seeing the natives recovering. Funding support, it's a pretty broad-based funding, and I always like to put up a slide, but it includes both provincial government, utility companies, local government, and a lot of in-kind assistance as well. This year also we did some aquatic surveys. So Christina Lake Stewardship's doing an awesome job here at Christina Lake, but we don't really have a Christina Lake Stewardship Society everywhere else in the boundary. So we've taken that on with some funds from the Ministry of Forest, Lands, and Natural Resource Operations, who's actually responsible for everything water. Anyway, so we finished up. Last year we'd done a number of aquatic surveys in lakes. This year we added to that and finished off what we figured the majority of the priority areas, survey to see what there is. So this involves canoes, kayaks, hiking. We did everything from visual surveys for New Zealand mud snails to sampling for aquatic plants, auditory calls for bullfrogs, and visual sampling for red-eared spider turtles. We did a little bit of plankton toads. Most of that happens out here in the lake with stewardship. So some of the lakes we sampled this year, we did 33 locations. We had finished off a couple of locations on the rivers, a couple of creeks, another 15 lakes, and a number of wetlands. So quite a few areas. So we didn't find, fortunately, the boundaries in a really good position. So most of the province, they actually haven't done surveys like this to even know what is around. And so now we have a pretty good baseline of information. We have not been able to find any New Zealand mud snails. That's awesome. We have not found any American bullfrogs. Really good too. We haven't found any red-eared spider turtles detected, although we know of some escapes that may possibly show up. So yeah, we're in pretty good shape for those. As far as aquatic plants go, we didn't find any new species. We didn't find any hydrilla. No, we haven't found any of those really nasty plants, flowering rush, any of those, which is really good. We did confirm a couple of private landowners about yellow flag iris and also fragrant water lilies. So, we found a few that we have in the area, but nothing new, which is good. What we also found was some giant Japanese knotweed on the petal. Giant knotweed, it almost grows up to the root. It's huge stuff. The leaves are the size of a dinner plate or bigger. So it's quite a plant, not Jen standing in amongst it. So anyway, we did find these two plants along the kettle, and this is a real concern. When knotweeds get into river systems, they spread quite rapidly because they spread by root fragments, and of course our high water, grabs some root masses, grabs some dirt, carries it down farther, and it starts to grow, and it creates these just huge barriers along the river system. We only found one location of each actually in the river. The one was introduced by someone dumping garden waste on a neighbor's property, and the other one, we're not sure how it got there. But we're fortunate in that we haven't started to see any hybridization between them, and we think that that's going to be it. There's a couple more spots we would like to survey, but overall, our river system is in really good shape. We didn't find very much rush olive here, so. Where we're going, I guess as far as education, we promote clean, drain, dry, and encourage people to don't transport fish. And next year, we are hoping to start getting into schools with the Don't Let It Loose program, which Brenda was already starting to use out here to encourage kids, mostly it's kids with pets, not to let them go. So because we don't have a lot here now, our focus needs to be on preventing introductions.
And that's some of the funding sources. So again, it's pretty, with aquatic species, it only certain partners actually fund the program. The regional district is a big funder, Canada Summer Dog. Midway and Greenwood have contributed, and I'm missing Area C, that's the wrong slide. Because I updated the slide rate. I had last night, I honestly did. Christina Parks and Recreation has contributed into the education program for the species survey, and the Ministry of Forests has done most of the aquatic plant survey. It was there, trust me. Okay. Grace is, she's a good partner. She's an awesome partner. So the provincial update. This is there's a lot actually going on right now, provincially and federally. I'm only getting little bits of information about things here and there. The province is working to establish a Western Canadian invasive species agreement between the province, feds, and the territorial governments, and they formed this alien species task force. So what this means is they're actually trying to get all their ducks in a row as far as legislation, funding, and how they're going to work together. That's actually a huge step. They've been doing this out east at the Great Lakes for a while, but now this is Western Canada that's happening now. Also, the BC Mussel Defense Program, which is those roving teams you heard about. The numbers we were given was that they did find 300 boats that were from high risk, or 70 were from high risk areas. 34 were decontaminated, 15 with mussels or larvae, whether dead or alive, and 12 of those were from Eastern Canada. And they did actually quarantine some boats. They held boats for a month from people. So they were serious when they were out there, which is really good. It's unfortunate that it's not mandatory, but the last, or like up all the time, what a presentation I was at about a month ago, they said they are looking at, as they do these roving inspection stations, they're trying to identify areas that might be suitable for permanent inspection stations. I don't know if that'll actually happen or not, but one of the things that was interesting, Midway, when they had the scales there, where they have the inspections, that had the most U.S. boats going through it out of all the places in BC that they had inspection stations. So maybe we'll get lucky in our area, and if they move through to permanent inspection stations. But Grace, you got to keep on them to make sure they actually do it. The other news right now is the Pacific Northwest Economic Region, which is, think of it as all your politicians, your high-level politicians, your premiers. What do they call them in the state? Governors, that kind of thing. Anyway, they're all meeting actually this week, and one of the things they're working on, they have a 2-day workshop on mussels and how they're going to deal with mussels. So this is a good step. I mean, things are happening. I don't know what all the outcomes will be of it, but it's good when our leaders are actually talking about the stuff and how to work together to deal with it. So that's my comment.
Any quick questions for Barb? Everybody's going to be here too. We need to make it quick because we're about 15 minutes to lunch. We have the presentation.
Are there any kind of pickles that are non-invasive in the rock next to the rock?
It depends on the kind of thistle. There's a couple native thistles, one that's usually higher elevation. I'm going to guess maybe the one you have is Canada thistle, but get me a picture of it. And if it is, we actually have biocontrol insects on it, so that is a thistle that doesn't become a huge problem, put it that way, because the bugs are doing a pretty good job. It takes them like 15 years, but they eventually do work. So we just got to identify the thistle.
And the other one is the turtles that I've seen that one, top end of the lake in the void up section, they're all
Yeah, those are the painted turtles, which is the native turtles here. So what you're looking for with the red-eared slider turtle that has a red hatch, or a splash, I guess you could say, right across the head, and it has a different color belly. Well, they all look shiny when they're wet. Yeah, different colorations on the belly. So the painted turtle's bright orange, I believe, isn't it? And very pretty. No, the native doesn't.
All right, one more to go. Take your 3 rounds of applause.
So we are really, really lucky to have with us Holly McCollum, and Holly is a Hubble Tribe, right? And you are the principal investigator, Fish and Wildlife Department, and you can do a presentation on the genetics and isotope study being carried out on the Upper Columbia Watershed above Grand Coulee. Thank you very much for coming and joining us, Holly.
Good afternoon, everybody. So I'm Holly McCollum. I work for the Columbia Community Department of Fish and Wildlife. And I run a few research projects, and one of them is studying wild protein and and I'd like to recognize my team that come up here today, Shane Holder, biologist as well, and our great technician, Robert and Coleman. A very popular wild protein fishery. They're made of a very large cytoplasm, 22 to 24 inches. Very large cytoplasm. Most protein fisheries are smaller than 12 inches. So, and then we also have a hatchery component to the Lake Roosevelt fishery, so the majority of the fish harvested are from the wild population. So our question is always, well, where are these companies coming from? Because they're not shoreline spawning on Lake Roosevelt, because it's drawn down 50 feet every year, so that would dry up the eggs and be high spawning. So are they naturally receiving the rest of maybe very deep spawning, but they have to be below 50 feet? Are they integrating in water upstream? One of the first things we set out to do back in 2000 was do a basin-wide genetic study, which means that we had to go identify all of the coastal populations above Grand Coulee Dam, and of course fish swim downstream. We want to identify each one of those and see if those are contributing to our coastal fishery with Roosevelt. So what we have here is the Caldwell Reservation. This one is Grand Coulee Dam. No, that's not. That's Chief Joseph Dam. This is Grand Coulee Dam. This is the uppermost extent of the Columbia River where natural fish can go to right now. So then we have Grand Coulee Dam, and then the Lake Roosevelt stretches from Grand Coulee up to the border. And so here we are. So what we have, back in 2000 we started coordinating with our Canadian counterparts and going up into the Arrow Lake, which are the older ones dots, or all the totally populated being tested. The Scoobies, which are the sea chops on the right side here, Meadow Creek, and then I think the Coconut Creek, tested. This year was the first year around the float can, and found two coconut populations in there that we added to our baseline study. Those have not been tested yet, so we don't know if they're native or if they were translated from another population. So also on this graph, those circles are native populations, triangles are native. And so here we are at Christina Lake. You guys have two coconut populations, probably already know. And so this winter, we sampled the shoreline population, and then this past fall, we collected samples from Meadow Creek, and we've already run the shoreline population, and those are looking native to the going series. And but my biggest concern is to compare those fish to Lake Watson coconut. They look like Lake Watson coconut, which is where the blue dot is. Lake Watson is in Bellingham, Washington, and that is where Washington has used Lake Watson coconut in almost all their lakes. And so that's what the triangles are. That's what we use in Lake Roosevelt, or Lake Watson coconut. Lake Watson coconut are also put in the Ponderay Lake and the Coeur d'Alene Lake. And so, and also in another lake, Sullivan Lake, and almost all other Washington Lake Watson coconut. So I wanted to make sure we compare those to those, and it's great that they're not Lake Watson coconut. So we'll be running your other standard creeks next year to figure out which ones those are. But the going theory right now is their Meadow Creek coconut, which is your native, the pink dot up there in the meadows. So, but then Lake Roosevelt still came out yellow, or they're yellow, they're different, they're genetically distinct from all those upriver populations. So either I'm missing one, or we have another population. And so I'm going to work with some of our partners here in Canada to try to go above the arrows and start sampling some of those coconut populations today. But if they're a native and they're genetically distinct and different from all those other coconut populations, then we need to start protecting them. You know, one of our theories is that maybe they were a historic sockeye population
that was, became a kokanee because the Grand Coulee Dam blocked them. They can no longer facilitate their national life history, and they reverted to a resident life history, which kokanee can do. So of course, most kokanee can become sockeye, sockeye can become kokanee. And also, the rainbow trout steelhead are the same thing. So something to think about for this group is rainbow trout are a rainbow trout, and if you're a steelhead, you're just a rainbow trout with the ocean, or you can be a resident rainbow trout. So a lot of people say, well, this is steelhead, we're going to introduce steelhead. You're not really introducing steelhead, you're introducing rainbow trout. What we have are native red dance rainbow trout. So most of the rainbow trout east of the Cascade Mountains are considered red dance trout. And we have a lot of, a variety of native populations on the Congo reservation. And so we are also doing a genetic study on those fish and kind of cataloging them for the future. So far we've done 2 distinct populations in the Sampoil River. And some of our other counterparts that are in research like Roosevelt are cataloging red bands in the Spokane drainage, red bands up in the upper the Kettle and some other tributaries that we didn't touch on yet. So as we move into the future and hopefully someday we have a natural fish returning to our area, we'll already have all the genetic diversity cataloged for these fish. So it's important to catalog your red band trout, and so that the managers can discuss and talk about what are the options for best stocks or reintroduction. Be very careful, because the last thing we want to do is introduce non-native fish into our area. Non-native plants and other animals are going to get you don't want any of the non-native red band or sockeye. So again, for this one, so we didn't really find our population that I was looking for. So I want to try continue to look north, but our going theory right now is that they're native kokanee like Roosevelt that are holding on somehow through a resident like they're probably not. So because I can answer our question, we started doing a microchemistry study. So what this is an isotope study. We partnered with Pacific Northwest National Laboratories, and we're looking at otoliths of fish. And what you can do is the otolith lays down a layer, just like trees lay down a frame or a year of growth. The otolith lays down a layer of daily growth of fish. And so in that calcium matrix, certain elements in the natural elements in the the otolith is the earbone of the fish. So usually, if you ever want to get someone else, you keep the head of the fish and yeah. And so anyway, so a lot of the elements, the rocks out in the environment, they have natural elements like barium, zinc, magnesium, uranium, lead, and cadmium. And so, and those types of elements will attach to the calcium matrix in the otolith. So we can look at the otolith and look at the different elements. So if the fish in the water is high in magnesium, or strontium isotope. And if the water is high in strontium, it'll put lots of that strontium isotopes in that particular layer. If the fish moves away from a high strontium location, those values will go lower. So we kind of look at movement of the fish. So my question is trying to figure out where are these kokanee spawning. If I try to look at the different strontium levels throughout the basin, I try to locate where maybe the spawning location is as well. So we started this study a few years ago. And part of this is to do a large mussel study. And so all these locations are where Shay and crew are out collecting mussels. And these mussels are the native Ogonata, the mostly silver type. And we did find 2 populations on the native pearl shell mussel, Margaritipra, which is really cool because it's very, very rare. And they live up to 100 years old. And so we found one population in the Sampoil River, and then one in the Kettle River. So it'd be really interesting to know if anyone else has seen these mussels, the western pearl shell, up in the basin or anywhere else. Those are the only 2 locations that we know of in Eastern Washington. And we have one of our partners, the U.S. Fish and Wildlife Service, they have a mussel experts. So if anyone ever has a picture of mussels or you don't know how to identify them, they're very good. Send us a picture too, and we can try and see that out. Oh, that is great, yeah. Okay, yeah, so yeah, whatever you do, just take a GPS point of where you are, and then you just so, oh, and then with water, so you take water samples for all your different locations. That's what the blue dots are, and that's the water growing to my ratio there, and the muscle and the fish try to happen. So, we use laser ablation, which is a nice fancy laser on that TNL trace. Cut the otoliths in half. In the very center of the otolith is the maternal zone. In the very middle center of the otolith is where the mother is spawning and where her otolith ratio. This is kind of interesting. So then as you move away from the center, it's the needle rearing area, and then there's the growth. So that's and how we look at that. So the water quality results were not as good as I wanted because what we have is this yellow, the yellow dots are Lake Roosevelt, the Columbia River, and it was all the same. It's all the average of 0.15 or 7.714 strontium isotope ratio. So there wasn't really a change. What I needed was a change in the ratio that the fish were going. That wasn't really helpful, but what was helpful is all west side rivers are very high in strontium, and also everything west of Lake Roosevelt on this side is high, everything on this side is low. So then we can kind of tell fish are coming from the Ponderay system coming in, or Spokane coming in, or if they're from Loops, say Christina Lake or the Tuttle. That was helpful. So now we're going to start expanding it. Okay, so after we ran everything, I was supposed to get one life history pattern of this cooking and figure out where they're coming from. Well, of course, that's 4. And so what happened was about 27% of those wild cookies came from a low strontium. Okay, so the middle of right here is the middle of the maternal zone, and then you move out. So this is the middle of all of the fish. So 27% came from a low strontium area that migrated to Lake Roosevelt, which is the red line. And then 36 came from a very high strontium area, which is way higher than any of our water samples identified. So that's another one where we are going to expand. Maybe they're coming from higher up in the basin and go try and figure that. And then a couple of these others have these kind of odd up and down ratios. So our future work is we're expanding, so we need to expand that water to try and find those high strontium ratios. So I want to go all the way up, okay, up into the Columbia Lake, and we're expanding east into the Ponderay system and the Coeur d'Alene system. And then we're also going to be expanding our elements we're testing. There's boron, and carbon, and some other elements to try and tell the between these different areas. And the code study. But another good application is now that we've invested all this money and time into the water and mussel study, we can use it for other fish. So we have that pipe, northern pipe issue in the upper Lake Roosevelt. So there was a survey done this past spring, the otoliths. I was able to run those otoliths with a strontium ratio. We already had that water quality mapped out. And so what we found in some of we only tested 5 local types, but they're coming from a very low strontium ratio area, which is Kettle River area. And then they were there for quite a while, and they made a fast migration to Lake Roosevelt. Kind of a similar thing here. They were born somewhere low strontium, and they migrated into Lake Roosevelt, which is an issue because we were assuming most of these fish are coming from the Ponderay system, coming down, and that's how they originally got there. But now they have established themselves in 2-year classes of pike, and we think that they might be spawning in the Kettle River. So now the next steps are to try and locate where they're spawning in the bays, of course, pike being shallow areas. But there is actually Spokane Tribe and WSW are leading up a pretty large effort, an aggressive effort to eradicate these fish. So there will be a netting program that they're facilitating this spring. And I don't know how many nets they're going to set. I want to say they're going to do week-long surveys, maybe 50 nets a day, and they'll fill all the pike areas. Try and get them the spawning season when they're up shallow.
And then also the harvest regulations on the road dog for piper. You have to retain every pipe you capture as an angler. So they're really trying to be aggressive with the situation. The last thing we want are these pipes to continue down the Columbia River into the Okanagan system, down into where there's endangered salmon. So anyway, I wanted to share kind of what we're doing with you guys and let you know that we're doing this kind of work. Maybe there's other folks up here and they can do similar work and share with the water quality mapping or utilize this resource for some other fish that you might want to look at. We're willing to share any of our data with you guys.
Thank you so much. Now that's what you call cross-border cooperation, right? I think there's a couple of quick questions out here. And are you holding the around so well so people can visit with you? Okay. Go ahead. Three-part questions. And there are fish ladders on the two dams. Are you in favor of it? If you're in favor of it, you know, what's the process for getting fish ladders in, and then what can we do then to do it?
So you're talking two dams, you mean Chiefdale and Grand Coulee? Right. Yes. There's some work in the States, and they're working with a variety of the funding agencies. So getting fish hatches is very, very expensive. And a lot of the questions surrounding it relate to, well, does this have higher flows? Are we having lost revenue through the dams? Or who's going to make up that lost revenue? So it's kind of a difficult situation. So they're coming out with some creative ways to try to get the fish over the dams, not just by fish ladders, which are very, very expensive, but by shooting them up. I don't know if you've heard of roof system, you know, and things like that. So there's some research going on to see if those types of fish are really successful. They're talking about truck and haul fish, marking the fish, track them. There's a variety of ideas out there. No one's been able to implement them yet because of the funding part of it. But I think it would be great for the economy to have salmon fishing opportunities. I think people would really enjoy that. And what we're doing also is that some of the historical papers I read said that there was a waterfall in the creek, so salmon couldn't make it back into the lake, and that's really one of the documents. But then when we were coming out talking to Brenda, I'm like, well, you have a barrier. She's like, no, we don't. I'm like, I read you have a barrier. She was like, you don't. So my great staff here, I said, hike the whole thing. Start and hike it, and then tell me, come back and tell me if there's barriers. And then we have protocol on how to measure, like, to determine if it is a fish barrier. And they hiked the entire thing. There's no barriers here. Well, okay, you know, so there's just a lot of groundwork things we're doing. So we're finding out, so we're trying to map out the barriers, where are the barriers, how much that survey to say, okay, we need to restore this creek, if we do, it might be able to produce this information. So there is a whole round of things going on. The fish ladder discussion, and there's a huge list, and I'm not really involved in that. I'm about answering questions that people have about how to get involved. There are people working on this. Yes, yes. There's a lot of great people working on this. Personally, of course, I'm.
This water has a wall on the east side. There's a huge shallow bay. Is that the primary area?
Yeah, up the hill, right to the right, there's that big shallow bay.
Also on the Columbia, on the east side, this upstream.
Yes, in the Market Flats area. Yes, that's one of the target areas. And in fact, last year, which was actually kind of a drawdown, maybe a good thing, they dropped the reservoir really fast, 20 feet, and dewatered the flats area, and they kind of 50 pipes in it, so because of the watering. So we don't like to dewater our shoreline, but that actually might be a good mechanism to kill these fish off quickly from watering. We've got a person there looking at us, so thank you, Holly.
Okay, so please enjoy your lunch. It is Christmas first, so you can say hi to Neil when you get back there. And by the way, if you don't know, that's Mary over there. And Mary's been helping out, and I will ask Brenda later on to introduce everybody that's here. So, going on the beach. And the door prize drum.